Hey guys, hope you're having fun in San Francisco. Here is what you need to be doing since you missed class. Look through all the pictures, take notes on what you don't have, and if the pictures are bad, blame Catherine because she took them. Don't tell her I said that. If you have any questions, please email me, but also come to tutorial on Monday morning when you get back so we can review stuff. The packet that you have that shows all the little pieces and it tells you to put stickers on things, I will see if I can hang on to those models until next week so you can use them. But if I can't, I can't. And you can just hear from your classmates what we did with those. So, protein synthesis. You have to start with DNA. Remember, central dogma, DNA, RNA, protein. DNA has all the instructions. So if that's this part, nucleotide, 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 and then you've got the mRNA because it's a messenger. So it's going to go into the nucleus. It's like if you go to the library and you have a book that you can't check out, but you can make a photocopy of it. That's the DNA. It has to stay in the nucleus, like that book has to stay in the library. So you make a photocopy of it that can leave the library. That's the messenger RNA. It's the message that goes. Then you end up with the mRNA leaving the nucleus. We'll talk about how everything gets edited and spliced because not all of it leaves. The mRNA goes and it provides the codons in triplets. So that's what that second packet is for that has the table on the top and the circle on the bottom. Those are the triplets that allow you to code for amino acids because you have to have 3 nucleotides to code for 1 amino acid. Okay, so take a look at your protein synthesis practice 1, and it kind of starts like this with the DNA and then it opens up and then it closes. That top part is your non-template strand. It's not the template that's being copied. The bottom one is the template strand that's being copied. Notice how it's all blank? You have to fill in what those bases would be based on what their complementary bases are on the non-template strand. So your first task before you do anything is to figure out what are the complementary sides of that section of DNA. Then you can figure out what the mRNA transcript is going to look like. So if you have the non-template, the template, and then you have the mRNA that comes in right here, you can then figure out what each of those nucleotides is going to be. And please don't forget that you have to substitute uracil for thymine. Then when you take a look at the codons, so remember the codons are the triplets, the 1, 2, 3s, because amino acids are read like that. So you have the codon from the mRNA, then you have the anti-codon, anti, opposite. So opposite the codon is on the tRNA, anti-codon, tRNA. And the tRNA, if you look back at that image that shows the aminoacyl tRNA synthetase and that enzyme that speeds up the process of linking together each individual amino acid with each tRNA that's going to carry it, that's why it's called transfer RNA. You can see that in order to bring the right amino acid, you not only have to have the tRNA that matches with the anticodon, that's why they know how to read the mRNA codon. Okay? So you might be wondering, how does the DNA actually make the right protein happen? Well, think about it. If you have your template strand of DNA, then you make your mRNA transcript, and then you have your mRNA transcript, And then you have your tRNA come in, that's the anti-codon. That tRNA is going to look just like the DNA did, but switching U for T. So you do end up having the same DNA expressed, even though you had to go to the mRNA and then to the amino acid language. You're going to get the same information that's on the DNA. Okay, last thing in terms of RNA processing. So you made the copies, you've got the 5' cap, you have the poly A tail, so that the mRNA that's inside that actually codes for things doesn't get degraded. They also help the ribosome recognize where to chomp in and actually start making the copies. Now, big thing with RNA processing, the introns stay in the nucleus. The exons get to exit the nucleus to be expressed. So during processing, you've got these SNRPs. That's how you say SNRNP. It stands for small nuclear ribosomal ribonucleoproteins. Sorry, small nuclear ribonucleoproteins. They form all together with other proteins to form that spliceosome, and that's what interacts with the splice sites at the end of the introns to actually chop off what needs to go. Then that can be recycled, reused again, and only the exons get to exit to be expressed. So you want to know what your homework is. The whole packet. So make sure you use your coding chart where it's either the box that has the letters here that says first letter, second letter, third letter. So if you're doing like AUG, find the row that says A next to it and go that way. And you know that's going to be somewhere in that row. Then look at the top where it says second letter and see what column it's going to be in. Now you have a box of 4 that it could be. Then look at the right side and see which of the letters U, A, C, or G, and then you can match it and see which one you're going to have. Do your best. Get as much of it done as possible, but whatever you do, please, 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 please come to tutorial on Monday because that will really help. That way you at least have a night to do homework and then come to tutorial on Tuesday to check in and make sure you got what we're talking about. And you can use the circle the same way, but start in the middle of the circle. So that's the one at the bottom of that front page of the packet. And so you start with the letter that you have there. So let's say it's U, UAG. So start with the U, and you know it's going to be in that slice of pie. Then move out, find the A, you know it's going to be in that portion of pie, and then find the G, and that will tell you what it is. If you don't know what that symbol is, turn the page a couple pages, and it might give you the key of what the symbol versus the word is, or you can look in your book, because it'll be in there as well. So to add a little fun, I'm actually filming this in my old track coach's office because we have a basketball game tonight, and I went straight from school to basketball, and I'm like, I gotta make this video. So for your feasting of the eyes and some laughter, here are some pictures of me that are on his wall. I mean, he's got pictures of tons of his athletes on his wall from when I was in high school. You ready? All right, there's me. Can you see it? I can't even tell if you can see it. Right there, and then right there, and 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 those are just a few shots.